rules, our agenda is to look at the rules and the procedures in the cell for how movement and physical activity takes place within a cell, right? And again, the onion example of sulfuric acid moving from the onion to your eyes is an example of something we call passive transport. Passive transport is movement, as you can see from the overhead. Let me mute everyone and march through. I'll turn on the chat room and we'll answer questions at the end. Let's see. Okay. So again, there are two types of movement within the cell and within the body. One type of movement is passive transport, the other is active transport. Understanding both of these types of movements are essential in understanding human health and also how things move in the environment and world around us. So I gave the examples a moment ago of onions and of blood pressure. Why does eating salt cause high blood pressure? Why do onions irritate your eyes? Both are examples of something called passive transport, which is movement of materials spontaneously, not requiring ATP energy in the body. Passive transport is movement of materials from a higher concentration to a lower concentration. What does that mean? What is concentration? Concentration is amount of stuff. So if my armpit does not, if I don't use underarm, which I did do this morning, by the way, if I don't use underarm deodorant, then the smell molecules are in a high concentration here, right? And then they will move out from my arm where there's a high concentration all around to where there's a low concentration. That is natural. That is normal. That is passive transport. That happens spontaneously, right? That means just without any input normally. If I said to you that all your smell molecules are coming through the computer and going underneath my armpit, you would say, definitely you're crazy. And I would agree with you. I'm not really saying that, but I am saying that that's the opposite, active transport. If everyone's smell molecules in a room come under my arm, then we say that's not normal, right? That's not spontaneous. That would require a lot of ATP energy. I suppose it's possible we could grab them and put them underneath and swish them. It would take a lot of energy. So that's the difference between active and passive transport. Passive transport, again, let me identify this. It is movement from a higher concentration to a lower concentration. And active transport is the opposite. Movement from an area where there is a low concentration, like all over the room, bringing smell molecules to a high concentration underneath my armpit. So again, one is natural, a passive transport. One is kind of unnatural, active transport. They both do happen in nature. So in a way, they're both natural. They're both what happens. But one happens spontaneously, passive transport, and the other happens not spontaneously, requires energy. So again, the example of smell, perfume, all of that is really due to the high amount of what we say KE. KE is kinetic energy of molecules. That's random motion of molecules. That's the reason for passive transport in the body. Molecules move away, away, away from each other, naturally hitting each other due to their high kinetic energy. On the other hand, I put an example here. The sodium-potassium pump. What does that mean? We're going to end the lecture with that today, but I want to give you a little highlight before we start. That's an example of an active transport system in our body. It maintains an abnormal amount of sodium on the outside of a cell and potassium on the inside of a cell. Why do I say abnormal? Because under normal circumstances, everything wants to become equal on both sides. But this pumping system, close your eyes, feel your hair, feel every part, feel your ears. Every cell is carrying out sodium, potassium pumping. The sodium potassium pump requires a lot of energy in your body to keep pumping out and making all of your cells relatively negative inside with a lot of sodium outside. So again, what should you know? Take-home message from this: passive transport happens spontaneously, does not require energy, and just passively moves molecules from a high to low concentration. Again, passive transport moves molecules from a high to low concentration, no energy needed. Active transport requires energy and moves things from a low concentration to a high concentration. Always anything that requires energy is active transport. You remember that from the word active, requires energy input. And here's a picture to help you remember. There's 3 types of passive transport: diffusion, facilitated diffusion, and, well, I guess 4 types: osmosis and filtration. We're going to go over the 4 types right now. Hold on to your seats, everyone. All right, diffusion, again, let me highlight it and define it. Everyone likes a definition, right? Diffusion is movement of molecules from a high concentration to a low concentration until there's an equilibration. In other words, a reaching of equilibrium, equal on both sides. So in your body, in your cell, alcohol, fat-soluble vitamins like D, A, E, and K. I can remember that because that's my last name, D, A, and E, and then just add a K. Those are the fat-soluble vitamins. O2, carbon dioxide, gases. All of these move from a high concentration to a low concentration across a cell membrane. And why is that? Well, because they're able to move through the membrane without the help of anything. Like dissolves like, right? We said that in the last class. If you're fat, you dissolve fat. If you're charged, you dissolve charged. So the membrane is mostly fat, right? The bilipid layer is mostly fat. So things that are fatty go right through a membrane. That you should remember. Commit it to memory, because fat hormones, fatty vitamins, D, A, E, and K, or little things like these gases I highlighted, oxygen and carbon dioxide, get through a membrane rather quickly. Alcohol, that's why alcohol hits you when you drink it. It's quick because it diffuses in relatively quickly. There's a good amount of fat base to it. The odd thought is a truck going down a hill, right? Here's a little hill. Diffusion is movement down a hill. The hill is called a concentration gradient. You can write the word a hill next to this, H-I-L-L. Concentration gradient really means, the word grade or gradient means difference in between 2 spots. So there's a difference in concentration. It's like a hill. So if you're going down the hill, it's called diffusion, movement from a high to low concentration. And your textbook's got a picture of diffusion, a type of diffusion in figure 3.24 and figure 3.25, all right? Shows you some examples of that movement. Facilitated diffusion occurs for chemicals that require the help of a carrier protein. And by that I mean, these chemicals cannot just get through a membrane that easily. Water kind of wiggles its way through, but it's a little bit charged, as you know from the chemistry lecture. It's polar covalent. Amino acids and glucose are a bit too big and a bit charged. So they require the membrane proteins. Remember what we call them? Integral proteins. Well, that's why I had you learn the cell membrane fluid mosaic model. Remember that from the last lecture? The fluid mosaic model shows integral proteins bobbing up and down in a phospholipid C in your textbook. And we call these integral proteins membrane proteins, also known as permeases. So I wrote the word permeases here. In a way, they're enzymes, right? ASE, they're enzymes that help things happen. They help to ferry, like a little boat, ferrying across the membrane, these different chemicals: sugar, protein, and water. And they have little channels in them as well that help. What is all this called? Facilitated diffusion. Why facilitated, you're asking? You know, facilitated means, what's a facilitator? A helper at an event, right? Well, this helps the event of moving things across a membrane. The third type of diffusion, of passive transport, is diffusion of water across a selectively permeable membrane. That is known as osmosis. When it's just regular diffusion of any chemical, you call it diffusion. When you add water, when you talk about water, you just immediately say osmosis. Osmosis refers to osmolarity, or concentration of solute in a solution. Solute is stuff dissolved in a solution. Solute is salt, solute is sugar. I made my coffee. I have sugar in it. The sugar dissolves. The sugar is the solute, and the water is the solvent. And the osmolarity refers to the amount of solute that's dissolved. So before you take a look at the screen, you right, everyone, you can see the screen, I'm sure. If not, comment to me. I got it all under control, by the way. Aren't you impressed? New operating system, zooming in, high efficiency for a guy who did his thesis on a typewriter. We didn't have computers back then, just so you know. But anyway, making a long story short, I'm in a good mood because things worked out. The last class was terrible. When the computer doesn't work, you know how irritating it is. By the way, I have some good news for you guys. I'm going to drop the lowest test. I don't know if you approve of that or not. I think you're kind of crazy if you don't approve of it, but I'm going to drop the lowest test because sometimes things don't work out. What if you can't be there? What if the computer doesn't work? I can't really give a makeup. It's almost impossible. So in lieu of a makeup, there's no makeups. I dropped the lowest. That's it. So one of the lowest. So either a practical or a test. You get to choose at the end. I mean, I'll obviously choose the lowest. So I'll just drop the lowest. So this will make it a little less stressful tomorrow's exam. And also I like doing that during the year. I always do that. And I don't have it in the syllabus, but I'm going to add it. And this way, you may be new to college. You've never taken a test. If you do lousy on the first test, I don't want you devastated. I want you to make the changes necessary. And online's a challenge in and of itself. I thought of that because yesterday it was terrible. I figured out what happened. Thank God for the microbiologist. There was just a minor issue, but this can happen to all of us. What if you drop your computer in the bathtub? So I'm dropping the lowest test. Now, I don't know why I'm doing an aside right now, but the reason is osmosis is our fundamental, most, how do I put it? Difficult topic in A&P 1 because people always get it wrong. So what I want to do is present this to you in such a way that you'll never get it wrong. Take a look at the 3 beakers in front of you, okay? Each beaker has a cell with a 0.9% osmolarity. That is normal physiologic saline. All of our cells are 0.9% sodium chloride. So you should know that, mainly because that's what you get shot up with every time you go to the hospital. They put an IV drip in, that's saline to keep you hydrated. And if there's a lot of stuff dissolved, we call it hypertonic. If there's very little stuff dissolved, we call it hypotonic. Many of you have heard this, but many of you have memorized this the wrong way in high school and even in general bio, I have found. So what you have to do is think of this in terms of diffusion. Always, 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 materials move from a high concentration to a low concentration. Passive transport. We've been through that now. All you got to do is apply that to the tonicity here. What will happen in each cell? What is the changes in tonicity? Tonicity meaning amount of stuff dissolved. And that word is used almost interchangeably with osmolarity, how much is dissolved. So let's do the first case in the middle. If the cell is isotonic to its outside world, that means the outside is the same as the inside. That one's an easy one to remember. If outside and inside are the same, it's isotonic. Iso means the same in Latin. If the cell has less stuff dissolved than the outside, we say that the cell is hypotonic, right? Is 0.9% bigger or smaller than 9%? It is smaller. So if you look at beaker number 1, 0.9% is smaller than 9%, right? 9% is bigger. So this thing was put, this red blood cell, we'll call it, was put in a beaker of salty water. What happens when you put a cell in a beaker with salty water? The cell will shrivel because while salt will enter the cell, right? Going from a high concentration outside to a low concentration inside. Water will leave the cell. I would write down and circle something known as a golden rule. Everyone's got to have a golden rule in life. You know, a motto. I hope all of you have a motto. And this is your new motto for today. Water follows salt. Just write it down, memorize it. I said, put a star, circle it, and write it right here. Water follows salt. Water follows salt. Here I wrote it in a little bit of a different way. Water follows solute, because technically it's anything dissolved. Water will always follow. So there's a lot of salt outside of beaker number 1, right? Water follows the salt outside, and so a cell that is hypotonic will shrivel, all right? And, right, if the cell is hypotonic, it will shrivel and shrink. That's known as crenation. If we look at beaker 3, that's the opposite. The outside of the cell has very little, less salt dissolved than inside, right? We say that the cell is hypertonic. That means it has more stuff dissolved. The word hyper, you know, I was a hyperactive kid. That means I had a lot of energy. Hypertonic means you got a lot of stuff dissolved. Hypertonicity, right? So this cell has a lot dissolved. You see the cursor? It has a lot dissolved inside and less dissolved on the outside. So salt will leave and go to the outside, but water will go from the beaker into the cell and make it blow up, all right? Because water follows solute. It's a lot of solute or salt inside the cell, and so water will cause the cell to expand and eventually explode. Our application of this is that a high salt diet is bad for high blood pressure patients. Like myself, always have borderline high blood pressure. That's probably because I like salty stuff. Anything salty, I enjoy. Some of you are sugar people. Usually I would do like in the class, have you raise your hand. Do you like sweet treats or do you like salty treats? Everyone's a bit different. I'm definitely a saltaholic. And why is it bad? You know, look at your veins in your arm. Just take a look at your veins and think about eating salt, you know, eating your Pringles potato chips. And, well, you absorb the salt. It goes into your blood vessels. What's going to happen? Water follows salt, right? That's why I had you circle it, the golden rule. And there's a little picture of water following into the salt from your body cells into your blood vessel. See that blood vessel? High NaCl. High NaCl means that there's a lot of salt in the blood vessel. If you add water to a blood vessel, or what happens to it? Pressure makes it go up. That's why it's bad to have a salty diet when you're a high blood pressure patient, obviously, because the water follows the salt in your blood vessel, dehydrates you, by the way, which is why you never want to drink salt water on a desert island when you don't have much water, because it'll dehydrate you even more. More water will leave your cells, go into the salty blood vessel, raise your blood pressure, and you'll pee it out, dehydrate even more. And that's what happens with patients with edema. Edema means swelling. If you have swelling, you take hypertonic solutions. In other words, these are types of salt solutions that literally pull water into the kidneys to remove more water from your body and prevent that swelling from building up. And that's a treatment, you know, Lasix, different types of water pills that will pull out the water from the body. All right, that's a treatment for edema. Last but not least, everyone, is filtration. Filtration is transport dependent on pressure, hydrostatic or water pressure. And, you know, during a lab, usually I will have a little filter and you'll put liquids through the filter and see which ones stay and which ones don't. Basically, separation due to filtration occurs based on the holes in the filter, how big the holes are. Little things come through, big things do not. We say the glomerulus of the kidney. That's kind of the filter of the kidney. It's a ball of capillaries that act in a way as a filter of the kidney, push blood pressure on the capillary wall, and some things get in and some things leave your body, you urinate it out, and some things stay. That's, of course, an example of filtration. All of our cell membranes, our blood vessels carry out filtration because there is pressure on the walls and some things stay in and others don't. That is passive transport, the 4 types of passive transport. Now I want to shift gears and I want to look at active transport. Active transport is movement from a low concentration to a high concentration. Figure 3.26 gives the sodium-potassium pump as an example. You're going uphill, right? You require stored ATP energy to move from a low to a high concentration against the gradient, up the grade, we say, from a low to a high concentration. And the image in the figure, it shows it better than my notes here. But my notes, look at the notes first and then the image in the textbook. The notes have a picture of this integral protein which acts as a excuse me, which acts as a carrier enzyme, you know, a permease, to move sodium, 3 sodiums out with every turn and 2 potassiums in with every turn of the sodium-potassium pump. That is the end, and I know that's on the test, that is the end result of the sodium-potassium pump. Every time the pump turns, where the little carrier enzyme points outward and then points inward, 3 sodium is removed from a cell and 2 K plus potassium ions are returned to a cell. So I repeat, sodium-potassium pump will pump in and out sodium, potassium, and there's another calcium pump. That's a pumping system. I'm not going to get into that now. We'll do that when we do muscles, but I'm repeating, the sodium-potassium pump functions. Now listen up, functions to pump 3 sodiums out of a cell with every turn and 2 potassiums in to a cell with every turn. What does that do? If you lose 3 good friends and you only replace them with 2 good friends, right? Sodium's positive, potassium's positive, right? Happy friends, think of it as good friends. You're essentially what? Losing 1 every time. You're losing 3, but you're getting 2 back. So you're netting 1 loss every time it turns. So you're making the outside what? More positive. More of your friends are outside than inside. Inside is what? Becomes more negative. And that's why all of our cells are what we say negative 70 millivolts. Sodium-potassium pump creates an electrochemical gradient. And by that I mean the electro part, everyone look on the screen, the electro part is that the inside is electrically more negative. The chemical part is that, you know, the chemistry, it's more, there's an abnormal difference in concentration of sodium and potassium. So there's a chemical difference across. More sodium outside, more potassium inside. This electrochemical gradient is like a water wheel that will turn as you allow the materials through, okay, and create energy and drive other cell activities. That's kind of a deep thought, but that's the reason why you can catch the dollar bill. Or anyway, you can move your fingers to catch the dollar bill. You can't catch the dollar bill. You do it because you have this stored sodium and potassium allowing all of your motions, like your hand to move, or your brain to listen and think about this lecture. All of that is a result of the sodium-potassium pump, ladies and gentlemen. It is not a, it's not just something to memorize. You got to think of it as happening constantly in all of your cells, where you're making sodium outside, potassium inside, and the result, everyone, the result is that you have an electrochemical gradient that drives all these other activities, like you closing your hands. That's active transport. So on the test, I'm going to ask you, what is the result of active transport? The inside is going to become more negative, right? And you're going to have more what? So potassium. And the outside, you're going to have more what? Sodium. So that's the result. And I describe it nicely. I really believe that on page 108 and 109. And there's a little picture. But better to discuss in person. Now, moving along, and we're on to bulk movement within the city. How do we move big stuff in the city? And figure 3.27 shows the movement through exocytosis and endocytosis. Exocytosis is the bulk movement of materials out of a cell, where materials go into a vacuole. Remember we talked about vacuoles as storage compartments in the last lecture? They go into a vacuole, waste, water, hormones, and they fuse with the cell membrane of the cell and pop out. Endocytosis is movement of bulk materials into a cell. You don't have to know about the clathrin-coated vesicles. That's getting too detailed here, so just cross that out. What do you care? But what you should know is the words phagocytosis, pinocytosis, and receptor-mediated endocytosis. Phagocytosis is the gobbling of solid materials, and your immune cells act that way, and many of your cells act that way. Figure 3.18 on page 99 is a picture of whatever, an amoeba, some creature gobbling up an enemy, where it's pseudopods or it's fake feet, we call it. Even our own cells do this. Our little fake feet surround and gobble up and pull in an enemy. That's how your immune cells work, and the lysosome digests it. Pinocytosis is cell drinking, when a cell membrane just invaginates and forms a little tube in, and you suck up liquid and dissolved material. And my favorite is receptor-mediated endocytosis, everyone. Receptor means it has a little structure on it. See the little structure? Everyone see the receptor? It's a specific shape, and it matches to a specific molecule. Can be a flu, like coronavirus. It can be an enzyme. It can be a hormone. It can even be fat. And the receptor specifically, look at my hand. It's the receptor, and my fist is the, let's just make believe it's an enzyme. They fit together, and that causes a change to pull in the material. That specific fit is referred to receptor-mediated endocytosis, and many of our materials get into our body specifically in this manner. I want to talk to people with high cholesterol. If you have parents with high cholesterol, probably you do, at least a third of you, because that's statistically the number. Often it's due to diet, but often it's also due to the receptors. The receptors for what we call your bad cholesterol, your LDLs, are not shaped right, or you don't have enough of them. So the bad cholesterol stays in the blood longer, and, fat builds up on the arteries, and that's bad. Stroke, heart attack. Hormones like insulin, very common example. You have to have enough insulin receptors, and if you don't have the right insulin receptors, that's diabetes, right? If you diabetes type 2, the insulin kind of can't attach to the hormone and pull in sugar, and that's why a person is not able to use sugar right. That's diabetes. So these are examples. Coronavirus, for example, you've got to have a shape for it that attaches to. And that may be why some people are more susceptible than others. It always comes down to the receptor-mediated endocytosis. Do you have the receptor for it? And that means you'll get either that flu, or you'll properly function getting rid of your fat in your body. I want to end our discussion of this with movement, a summary of movement across the membrane. Way in which we move across a membrane depends on the distance. How far are you between the blood vessel and the cell? Everything is pretty close together in the body. What's the size of the gradient? The higher the gradient, in other words, the steeper the grade, the faster you move. Molecule size. If you're smaller, you move faster. If you're smaller, you move faster. And temperature. If it's warmer, you move faster. An example is on a hot subway car, things smell a lot quicker than on a cold subway car, right? Things just move faster. And what moves? If you're smaller or if you're fat soluble, you get through the membrane pretty easily. Once you're polar, in other words, you're a charged particle, or you're bigger, then you're not going to get in as easily, because the membrane is made of a layer of fat, and polar things can't get through it. Many of you learned in high school about cell division. And, why put cell division in this chapter? Well, we have to understand how cells divide. You have a cut. If you cut yourself, if you grow, your bones grow. If, all these examples are cell division. You have to replenish the cells you have or grow new cells. And that is all called the cell cycle. Changes a cell undergoes from the formation to its reproduction. And there's really two phases of a cell's life. The cell cycle is really the cell's life. One phase is the interphase. That's the growth of the cell. Not much is happening. You're just growing. 90% of a cell's life is in the interphase. Makes organelles, doubles the genetic material, makes the doubling the amount of the organelles, let's say, makes materials for mitosis, breaking apart the cell. The cell division itself is prophase, metaphase, anaphase, telophase. And I remember learning that, PMAT. Teacher in my class, it was high school, said, Peter meets Anna tonight. Oh my God, I was devastated because there was an Anna in the class. You're so embarrassed. And it was really, I'll never forget it. And this is how, I bring it up partly to embarrass myself, partly to tell you this. That's the way to remember things. All right. Try to find little devices. We call them mnemonic devices, little ways to remember things, make a sentence or make a word. PMAT. Now you're going to remember that. These are the phases of mitosis. Prophase is when the nucleus kind of gets ready for cell division. Chromosomes smush together, all right, and get organized. And that's your genetic material. Usually it's strewn about as chromosomes. Excuse me. Usually it's strewn about, like laundry in your room. If you're not cleaning up, that's your genetic material when you're not dividing. It's in the form of what we call chromatin. You can remember that because it's thin. Chromatin is thin. It looks thin. And then if you bundle all your laundry together, right, like the cell bundles all its genetic material together, it's chromosomes. Nucleus disappears, and it's ready for division. Metaphase is when you literally see the chromosomes lined up in the middle. And anaphase is when the chromosomes pull apart to opposite ends of the cell. And telophase occurs when there's like a pinching, if you look, a pinching apart of the parts of the cytoplasm of the cell. And the actual division of the cytoplasm is known as the word cytokinesis. So why is this important? Why bother going over this? We're going over it because to understand cell division is to understand growth, like bone growth, but also to understand and all growth of all parts of your body, and also repair of your body. Additionally, I want to talk a little bit about cancer, which is the second leading cause of death in the world. Again, mitosis, easy to remember if you think of it as a date. In the beginning of a date, right? The prophase, things are all over the place. You got to start cleaning up your room. No one wants a dirty room, right? So you clean it up. Metaphase, you're in the middle of the date. Anaphase, Anna is running away from you because you're talking too much biology. And telophase is the breakup, the telephone call. Now, you guys don't have a telephone call. You guys probably text each other with the breakup. Telephone call was always awkward. It's not you, it's me, very awkward. Dead silence. So thank God for texting, right? Just text and be on your way. Much better. Little entertainment here. All right, before a serious topic. And we're going to end up with the serious topic of cancer, okay? Cancer is uncontrolled cell division. If you have cancer, that means there's some sort of uncontrolled cell division. There's over 200 types of cancer. Some are deadly. All are, let's just say, all are deadly if left unchecked. There are a couple of exceptions, like basal cell carcinoma, certain type of skin cancer. It just grows and doesn't kill you. But mostly, some are very treatable, and some are not. And so when we talk about cancer, we talk about 200 different separate diseases. And within them, many more types of diseases. It depends on the stage, how far it advanced, and it depends on how aggressive the particular type of cancer is. So what I'm getting at is cancer is uncontrolled cell division, and that is its definition. It results in some sort of tumor or growth, all right? It's when the phases that we just talked about do not happen in their proper order. So let's talk about number one, four characteristics. So I want you to memorize those 4 characteristics. Number 1, loss of contact inhibition. Usually, you know, a cell contacts another cell and it inhibits it. It stops growing. Not with cancer. It touches all sides of another cell and it keeps growing. Loses the ability to inhibit based on contact. Undifferentiated. You know, I always thought more cells, better. You know, why not have a kidney tumor? More kidney for you to function. The trouble is it's undifferentiated. That means it's not specialized. You can write that next to the word undifferentiated. Not specialized. Cannot do what a normal cell does. Next to the word undifferentiated, not specialized, that's the reason it's trouble. And then loss of cell affinity. I'm going to skip this immortality for a minute. Loss of cell affinity. Cell affinity means you like being with your own. Kidney cell likes being with kidneys. Liver cell likes being with liver, but not for cancer. That's why it spreads around. All right? So a benign tumor doesn't spread. If you have something that's benign, it just grows and can get in the way, and you do have to get rid of it sometimes, not always. But malignant, you always have to get rid of. Spreads to other sites. Somehow you have to contain it, get rid of it, you know, something with it. Usually surgery to remove it. Or radiation that we talked about 2 lectures ago in chemistry to destroy it. Somehow it's got to be destroyed. So again, let me reiterate the first 3 characteristics of cancer. Loss of contact inhibition means you don't stop growing when you touch another cell. Undifferentiated means you're not able to do anything. You're incompetent, unspecialized. And then loss of cell affinity, which means that you spread. Finally, immortality. This one I saved for the end because it's so creepy. Given enough food, a cancer cell will live for eternity. It will live forever. That's a creepy thought because it kills you, but it can live forever. It has the ability of the fountain of youth, a cancer cell. And they have the Immortal Life of Henrietta Lacks is a book about a lady, I guess she was about in the late 30s, maybe 40, had, I believe, cervical cancer, and they saved her cells, and she died, of course. And the family was pissed because they've been making money and you know, doing research on her cells without the family even knowing it, and they never reimbursed the family or anything. So, you know, who owns your own cells is the question. So it's a good book to kind of think about these things. Last but not least, we're going to do our clinical terms, and then I'm going to open up to conversation, talk about the test, and move forward. Anaplasia is a word. If you see that word, that means abnormalities like cancer. The cells look undifferentiated. They don't look normal. Next week's lab, you have the histology lab, and you're going to see what normal cells look like. When a cell is cancerous, it doesn't look like that. You know, it just has a little nucleus, little dots, very different. Words like hypertrophy and atrophy refer to muscles. Hypertrophy means increasing the size of a muscle. You know, you're doing your curls, making your muscle bigger. Atrophy means you're not exercising. You have a decrease in your cell size. Hyperplasia refers to increasing the cell number, but they're normal. So like thyroid cells, a goiter is like a swelling of the thyroid in your neck here, and right over here. And the goiter, I don't have a goiter, but a goiter means you're making more of more cells, but they are functioning right. And dysplasia refers to some sort of change in the cell due to an inflammation. It's not cancer, but it's, you know, an abnormality or a change. Now, it's been a good amount of material. We've gone about 45 minutes, and I think we should open it up to questions. Let's open the chat room, and let me open you up to all questions. Allow participants to unmute themselves. I think I want you to be unmuted. I want you to chat in the chat room if you have questions. And tomorrow at 9 o'clock, this will scare you into talking. You got your test. So, you know, if you have any questions on it, now's a good time. And we're done with the material. And next week, just so you know, we're going to start with histology. We'll have a lecture Tuesday at 9 a.m. on histology. By the way, if for any reason something fails with me technologically or I can't be here, I will email you and tell you that. I mean, I don't anticipate, but it's possible. I'll put it on the YouTube channel. It seems to be uploading nicely now with my new system. And I do not like just to upload a video and not have the interaction, okay? So I generally don't want to do it. But there may be a day where I have to actually head somewhere, and then I'll do that. I want to keep us to 9 o'clock, you know? Oh, and another thing I was going to say, you've got to buy the lab manual. I had one person ask, can they not buy the lab manual? You have to buy the lab manual. Somehow you've got to get it. I can't tell you to do it illegally and get screenshots from each other. That's not good. You know, you should have the manual. If you don't have the manual, you're going to fail the practicals because you got to have it. That's where I test from. Those pictures and those images and those procedures. We have a question. Will anything from the labs be on our tests or just our practicals? You know what? That's an excellent question, Taylor. I never put things from the lab on the exams. They only stay with the practicals. But I do put the 52 regions. There's 1 or 2 questions on the test from the 52 regions. You know, buccal, cervical, antebrachial, you know, all of that. Okay. You've got to, you know, antebrachial. That is the only thing that is going to be on the lab, and the reason from the lab to the lecture test. Usually, I want to keep lecture and lab separate. And then it says, Tanya, for tomorrow's test, we log in and you would verbally tell us the questions and we write them down, or you'll provide the exam just like our lectures. Don't log into Zoom tomorrow. I repeat, I'm not going to be on Zoom. Don't log into Zoom. I'm going to email you your test, and you're going to email it back to me within 50 minutes. There's 40 questions. I told you from now on, it's going to be number of questions plus 10 minutes of panic time. So I'm only kidding about that, by the way. You shouldn't panic. It'll all be there. You can look it up. I don't want to verbally tell you the questions. It's tempting, but I think we're just going to email you the test, and there's an answer sheet at the bottom. You know, just cut and paste your answers and the answer sheet, and then email it back to me within 50 minutes. I mean, there's a lot of ways to do this. I could use Vancohol and you log into that. I could verbally tell you and then watch you do it. But I feel like you should simply do, we'll do it this way. It's the most efficient, and it's, you know, an open book. I don't love open book, by the way. This is something I'm thinking about. I mean, I can't make you not have open book because it's online. I mean, I can tell you not to, and then some people will cheat and some people will not, and that's bad. So we're going to keep it open book, and we'll just limit the time. That's why. So, you know, have your notes out, have your textbook out, and you'll be familiar with it. I don't think it's bad, because as long as you're familiar with these terms, like I'll ask you, which of the following is, you know, is cancer? It's going to be multiple choice, true, false, and a little bit of matching. You know, which of the following is cancer? Anaplasia, hypertrophy, hyperplasia, dysplasia. You still have to know where to look at it, and you still have to kind of be familiar with everything. So that's important. So I'm comfortable with that. 40 questions. Just send me the answer sheet, and I'll try to grade it. You know, I've got to grade it by hand. I haven't found a better method. There's a lot of software out there, a lot like Zip. I do use during the year. It doesn't really work, so I'm not crazy about it. So, you know, as the teacher, I'm giving you the teaching techniques. Maybe one day you'll teach online, who knows what. There's a lot of routes to go. I like being with you in person. It does require us to commit to 3 days a week. Some teachers are not meeting with their students. They're just putting it up on Vancohol. I use YouTube. I don't like Vancohol. I use YouTube. This way it's open, And but I like meeting with you 3 times a week. But again, if you like, let's say you cannot meet, it's on YouTube, okay? So I try to keep it as flexible. I'm trying to make this as flexible as possible for all of us. It's more about getting, knowing the material, getting the material understood, and that's about it. So I don't want a lot of crazy, how do you put it? I don't want a lot of surprises for you. So that's why I'm dropping the lowest. If you have any, I don't think anyone's against that. It's the lowest test or practical, not both, just one of you have 5 assessments, 3 tests, 2 practicals, 1 is getting dropped. So I take the other 4 and I divide by 4. They're all will be worth 25% a piece. So you don't have to really ask what's my grade to date, just add it together and divide by however many tests you had. So if you have 4 tests, don't ask me what's my grade to date. Add up the 4 and divide by 4. But it does make me think, if you do well in the first 4 things, you don't have to take the last thing, right? It's kind of a thought. That's positive. What other goodies? Other questions for me? You're able to unmute yourself, right? I'm not going to force you to talk. Some people don't like a lot of chit chat. Yeah, let's see. What other goodies can I tell you? I think that's about all I have. I made the lecture a little shorter, so we didn't have to go crazy. Are you comfortable? You know what to read. You have these lecture notes. 9 o'clock, I'll email it to you. Okay, we won't talk. I won't be on Zoom. You're just going to take your test, and then 50 minutes later, you get it to me. I'll tell you, I urge you not to cheat with each other. Do not send each other answers, because gossip gets around, and then I have to bring you up on charges. Don't do that. And also, you never know who's giving you help. That's a mistake. You want to do it yourself so you feel comfortable and confident. I took driver's ed online, and actually my driving went downhill after that, but that's another story. But I liked it. I learned a lot, and you get what you can out of an online class. You're learning the same material as you would in class. Exactly. You may not be physically putting stuff in a bag in this week's lecture and in this week's lab, but you're still seeing the same stuff and I'm assessing the same stuff. So, it's a lot less headache for you and me in terms of a lot of driving. So this is the positive. And, all online classes have their pros and cons. I'll leave it at that. I could have sent you home a kit where you dissect stuff and, like this week's lab, you'd have a little bag and then you'd put stuff in a bag, in a cup and weigh it. But, it's, I don't know how much you're getting out of it. So I've been debating it. What other goodies? I don't know. If you don't have other questions, you can always email me questions throughout the day. Then 9 o'clock, I'll send you the test. Are you comfortable with this material? We could try a poll. I don't know. Maybe wait a minute. I don't know if I want to start polling you. We could do, I don't know. You want me to poll you? Do a poll for a second? A type of question. Let me just think. Nah, I think that's a waste. I don't think I'm going to do the polling technique. It's too time consuming. You know what? You can kind of make your own questions. I could actually make a question right now. Let's just shut this down. Let me give you a question verbally. Why waste it? Why waste time? I'll just tell you, a type of question might be, I'll put it in the chat room. I think that's easier. Let me think. Go back. All right. And you can, I know there's a matching column on this stuff, by the way. Make sure you know it. Which one's the powerhouse, which one's the capital, which one transports materials, that type of thing. the different junctions. All right, so one question might be here. I'll chat it out. Which best describes? I'll put it in the chat room. Okay, which best describes the cell membrane? And then I might say A. Which word? Static. B. Dynamic. C. Unchanging. D. Solid. What would you say? I don't know. One person give me their, write what they think. Put it in the chat room. Let's see if you get it. Could be fun. All right. Matt Murphy, B. All right. I get the drift. Everyone wrote B. You got it right. Don't be showing off, Madeline Greiner. All right, very nice. That's right. I like this. Yeah, I think that's a question on the test. It's not static and unchanging. It's not solid. It has little holes. It's constantly moving. Stuff like that. I think you'll be okay. I mean, I wish we would have it during. I wish we would see each other face to face. To be honest. All right. Can you go over the cell junctions again? That's a good one. Yes. On the test, why don't I give you a question? Which cell junction welds together cells of the uterus? And then I'll go over all three. All right. A, we're going to do a tight junction. B, gap junction. C, desmosome. Or D, ligand. I made one of them up, by the way. Why don't you try to guess it? Which welds together cells of the uterus? Yeah, Sky, you got it right. And let me show you, and it's a good question. Let me show you why. I got to show you the picture. And basically a spot weld. You've got a lot of intermediate fibers sticking out of the desmosome to hold together cells, okay? The gap is a communicator. See the little tunnel? That communicates. So that wouldn't be the answer. That's a communicator. A tight junction, that word is seals. It seals, prevents leaking. If you hear the word sealing or leaking, like leaking from the kidney or the digestive tract, it's tight. And it kind of makes sense, right? Like tight is tight, keeps it tight, seal it tight. Like, a diaper, seal it tight. So something like that. So anatomy and phys, very sensible. Everything makes sense if you think about it. Then again, there's a lot of words. My father says, what are you doing to these people? Desmosome, antibrachial. You know what it is? It's a language. It's a language, and that's the biggest problem with it. And just familiarize yourself with it. You should be all right. Any other questions? Always better to do the questions now than in an email. It's hard online, because, in class, I could, force you down and answer, hey, absolutely, you can't leave the room unless you answer this. I usually line people up, do like a gatekeeper. The few of you that have taken my class, that was like a gatekeeper. So you're forced to answer. It's a little fun, now that you've taken the class, it's more theater than anything to get you to study. So now you got to be a little more disciplined, everyone. If you're in class, I stand there, and then you get lined up and you get ridden the riot act. Hey, how come you don't know it? Now I can't do that. But I think you'll be okay. I think it sounds, as a group, like you're studying. I get your emails and you're studying. We'll see. And I dropped the lowest, so it should be all right. Keep me posted. Any other questions? Our hour is coming to an end, but I'm glad we didn't push together the cell bio for both. That would have been just too much, That would have been too much. I think cell has to be 2 separate lectures. It's just more than that period of time. Histo is going to be some time. Is it going to be a bit difficult, the first exam? I don't think so. I think you're going to be okay. Junie, I think you're going to be really fine. Those of you who've taken the class, it's a brush up, okay, from the areas that you were weak with. So only you know where you were weak. If you hated histology exam 2, then that's where you really got to study. As long as we study the notes, you should be okay. Sky, that's correct. I really believe that. The textbook is a strong and important backup, especially, ladies and gentlemen, because we're not together. We're in a 2D world. You need that textbook to give you backup. But I test from the lecture notes. As a matter of fact, I may sit here right now and do your practical, get that over with. And what I'm going to do is go from the manual and from the little handouts I give you, you know, each objective, I'm going to make your practical up. I think it's only going to be 25 questions. We'll do like a 35-minute practical. Get that out of the way. And that's it. I'm just going to write it up. And then we'll do the practical after next week. Next week, I want to do histo, I want to do the immune system, and I want to do the skin. I want to get those 3 done, and the following week will be your tests, all right? Next week, I'll give you the histo lab. And so next week, we're going to have no tests. But the following week, we're going to have exam 2, and we're going to have the midterm, okay? So let's get this test out of the way tomorrow. Next week, we can relax and just go through the 3 lectures, and I'll email you the histo lab, and that's it. Yeah, as long as you're studying the notes, should be okay. Are the practicals multiple choice? Absolutely. I'm going to make it up. True, false, multiple choice. Probably not a lot of matching, but you never know. That's what it'll be. I think you'll be more relaxed once you do your first test. You'll see, and you'll get confidence. But I think everyone, if you do well in your first 4, still take the 5th test. I mean, after all, you're paying for this, right? That's what I say. Okay. Last questions? All right. I will email. Don't go to Zoom tomorrow at 9 a.m. I'm going to email you the test and email it back by 9:50. Okay. I'll email you 9 o'clock, and then we'll see each other again on Tuesday, 9 o'clock of next week. All right. Email me if you have any questions. Bye.